Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International GCSE Biology Paper 2B for June 2022. This is the Part 1 video. I'll put the link to the Part 2 video below the description box. Let's begin with the first question. The first question has an essay that we have to read. So they say, supercharging plants to reduce global warming. The proportion of carbon dioxide in the atmosphere has increased in the last 100 years. In 2020, a mass of 727 gigatons of carbon dioxide was released into the atmosphere from natural processes, along with a mass of 37 gigatons from human activities. Scientists have estimated that plants naturally remove a mass of 746 gigatons of carbon dioxide from the atmosphere every year. The difference between what is removed and what is released causes atmospheric carbon dioxide to rise every year. Carbon dioxide is a greenhouse gas, and a significant rise will cause global warming. To help solve the problem of rising concentrations of atmospheric carbon dioxide, scientists are planning to produce transgenic supercharged plants that can remove atmospheric carbon dioxide and store it in their roots. The scientists estimate that if these plants can be developed, the plants could remove a mass of carbon dioxide equivalent to 50% of the emissions from human activities. Coastal plants that have their roots in seawater contain a substance called suberin in the cell walls of the outer layer of the roots. Suberin is a waterproof substance that contains a high proportion of carbon atoms. Suberin is decomposed very slowly to remain in the soil for a long time. The photograph shows a coastal plant called a mangrove. So this is the mangrove plant. To produce a supercharged plant, scientists intend to take the gene that calls for high suberin production from a coastal plant and insert it into crop plants. The crop plants use a perennial plant. Perennial plants live for many years rather than dying each winter. The transgenic crops would take in large amounts of carbon dioxide and use the carbon atoms to make suberin. The carbon would then be locked up and stored as suberin in roots. After successfully producing one plant, they will use micropropagation rather than pollination to produce others. These supercharged crop plants may have other uses. Suberin in roots helps to make them tolerant to soil with a high salt concentration, helping to produce higher crop yields in areas that have difficult growing conditions. The first part says carbon dioxide is a greenhouse gas. State the name of another greenhouse gas. We have to refer to line 7. Of course, another greenhouse gas can be methane, water vapor, oxides of nitrogen. The next part says calculate the increase in mass in kilograms of atmospheric carbon dioxide in 2020. We have to refer to line 2 to 7, and they want us to give our answer in standard form. When I referred to line 2 to 7, I saw that the carbon dioxide release in 2020 naturally is 727 gigatons, and that release due to human activity is 37 gigatons, and the total is 727 plus 37, which is 764 gigatons. Also, carbon dioxide removed was 746 gigatons. So to get the difference, which should be the increase, so it's going to be 764 minus 746, which gives us 18 gigatons. And since they've told us 1 gigaton is equal to that, I had to convert this into kilograms. So 18 times 10 power 12, that gave me 1.8 times 10 power 13 in standard form. So that should be the answer. The next part says state 2 consequences of global warming for the environment. Global warming is going to cause an increase in temperature, so that global temperatures are going to be higher, leading to the melting of the ice caps. And when that occurs, a lot of water is going to be flowing into the ocean, or the volume of the water in the ocean is going to increase. So a lot of flooding will occur. There will be loss of habitats because some land animals may not be able to survive where they are due to flooding. Extinction of some species. There will be coral reef bleaching, desertification, spread of pests as some migrate from different areas to other areas. Any of these would earn you the marks you're looking for. Let's move on. Here they say explain why producing genetically engineered plants with additional suberin in their roots could reduce atmospheric carbon dioxide. That is line 14 to 17 and line 19 to 24. So because this is genetic engineering, of course, these plants are going to take up a lot of carbon dioxide through photosynthesis. So I said the plants will take up carbon dioxide through photosynthesis, and the carbon dioxide will be converted into suberin and stored in the roots. Remember, the perennial plants grow for many years, they will continually absorb carbon dioxide for longer periods of time. You remember the passage said that suberin takes long to be decomposed. So this is going to be an advantage because the CO2 that was absorbed is going to be held into the suberin for longer periods of time without being released back into the atmosphere. The next part says which enzyme is used to remove a gene from a section of DNA. Of course, this should be a restriction enzyme. The answer should be a D. Part D says explain why the traditional suberin in the cell walls of the roots will make the transgenic plants tolerant to soil with a high salt concentration. This is line 26 to 28. I said using suberin with waterproof properties prevents water loss as the water moves from a high water potential inside the roots to a region of lower water potential, which is outside the roots, or you would say in the soil which has a high salt content, through the process of osmosis. Continuing on. Give 3 reasons why the scientists use micropropagation to produce the transgenic crop plants. Line 24 to 25. Micropropagation produces genetically identical crops, so we can be able to obtain large numbers of crops within a shorter time, and all produce crops will have suberin within them. And then problems associated with cross-pollination could be avoided because, as you know, cross-pollination can lead to differences in DNA or can lead to changes or exchange of genetic material. And that would mean that the crops that we would produce by those methods may not be 100% identical. So micropropagation is going to be a suitable method. This brings us to the end of question 1. Let's continue to question 2. Question 2. A student uses this method to investigate the nitrogen cycle. Take 2 samples of soil, each of mass 100 grams. Sterilize 1 sample of soil by heating at 100 degrees Celsius for 1 hour. Place the sterilized and unsterilized samples into separate filter funnels and pour 25 centimeters cubed of water through each soil sample and collect the filtrate in a test tube. Test each filtrate for nitrates. And then you pour water through each soil sample for 5 minutes. Pour another 25 centimeters cubed of water through each soil sample and collect the filtrate in a test tube. Test each filtrate for nitrates. So next they say add 1 centimeters cubed of a solution of ammonium salt to each soil and leave for 3 days. Pour 25 centimeters cubed of water through each soil sample again and collect the filtrate in a test tube. So before we go into any question, I want us to look at this sample. So we can see they began by heating a soil sample at 100 degrees Celsius. So this I'm going to say for sterilization. So meaning there are no microorganisms. So going down here, they say test each filtrate for nitrates. The diagram shows the student's apparatus. We're going to look at how this was done. So we see there is a funnel. The soil is going to be on top of a filter paper within a funnel. We see a test tube and then the filtrate that passes through. So continuing on the next page. Here they say the table shows the student's results. There is unsterilized soil as well as sterilized soil. So here the study said at the start of the investigation, we see results for nitrates. They were present in both. And then after water has passed through for 5 minutes, we can see both of them did not have nitrates. That means the nitrates have been dissolved or they've gone through with the filtrate. Then 3 days after adding ammonium salts, here we can see there nitrates present in here. It means microorganisms remained in the unsterilized soil, so they were able to find the nitrates present, while the one that was sterilized or the microorganisms were destroyed, the ammonium salt was now converted into nitrate. Give the independent variable in the investigation. The independent variable is presence of bacteria. The next part says suggest why the student poured water through the soil sample for 5 minutes before adding ammonium salts. This was to make sure that any pre-existing nitrates can be dissolved or they can be removed. Remember, all nitrates are soluble in water, so it means if there is a soil sample that has nitrates and you pour water, as the water goes through, it's going to dissolve the nitrates and the nitrates will pass through. So I said, to remove any pre-existing nitrates, this ensures that any nitrates present after the experiment must have come from the added ammonium salt. Comment on the results of the nitrate test on the 2 soil samples 3 days after adding ammonium salts. Remember, we had 2 soil samples. One was sterilized and the other was not sterilized, and we expect the unsterilized soil is going to have bacteria that could lead to formation of nitrates. However, the sterile soil is not going to have bacteria, and therefore we will not form nitrates at the end of the 3 days. So here I said nitrates will be present in the unsterile soil, while they will be absent in the sterile soil. And sterile soil will have nitrifying bacteria, and the nitrifying bacteria will convert ammonia to nitrite and then to nitrate. This brings us to the end of question 2. Question 3 says, the diagram shows a single-celled organism called Chlorella that lives in fresh water. Chlorella has a chloroplast and can photosynthesize. In this organism, there is a mitochondrion, a cell membrane, chloroplast, nucleus, as well as a cell wall. Which of these groups of organisms contains Chlorella? So we can see that this has a chloroplast, so it is not an animal cell. And also we see there is a nucleus, so this cannot be a bacteria. So this is another bacteria, and it's not an animal cell. When we look at the plants, we see there is no vacuole. In a plant cell, there should be a vacuole, so there is no vacuole or permanent vacuole. So this is not a plant cell. Therefore, it should be a protoctist. The answer should be a D. Moving on, they say, which of these labeled structures would also be present in an animal cell? So here, of course, an animal cell cannot have a chloroplast. That is out, this is out, and it cannot have a cell wall. So it should be this answer. It should have a cell membrane and a mitochondrion. That should be a B. Next, they say, complete the balanced chemical symbol equation for photosynthesis. This should be 6 carbon dioxide molecules, 6 water molecules, forming glucose, as well as 6 oxygen molecules. Moving on. So here they say the graph shows the effect of light intensity on gas exchange by Chlorella. On the vertical axis, we see there is oxygen released by Chlorella, and we see volume of oxygen exchanged in 5 minutes in millimeters cubed. We can see, of course, this scale goes below 0, so we can see there are some negative values. The x-axis is light intensity in arbitrary units. The first part says, explain why Chlorella takes in oxygen at light intensities below 10 arbitrary units. Chlorella can carry out photosynthesis as well as respiration, so we would expect that if there is less light intensity, there is going to be less photosynthesis, but there is going to be more respiration. So I said, at less than 10 arbitrary units, there is less light, so less photosynthesis takes place compared to respiration, and there will be a net gain of oxygen. Light intensity can determine if the rate of photosynthesis is going to be higher than the rate of respiration. When the light intensity is lower, there will be less photosynthesis, and therefore more respiration will take place, meaning more oxygen is going to be gained. So we see there is going to be a net gain in oxygen than the oxygen released. However, if photosynthesis is higher, or the rate of photosynthesis is higher than the rate of respiration, then we'll see less oxygen gain and we'll see more loss. So let's continue on. Here they say, explain the changes in the volume of oxygen released as the light intensity increases from 10 arbitrary units. If I take you back to the graph, we can see from 10, if it begins to increase, it means there is enough light or significant light to cause more photosynthesis to occur, or to cause a higher rate of photosynthesis than the rate of respiration, and therefore we will see less oxygen being gained and more being lost. 10 arbitrary units of light intensity, the rate of respiration and photosynthesis are equal. 0 oxygen is exchanged. That is, at this mark here on the graph, this point here. But as light intensity increases, we'll see a change in the volume of oxygen that is being exchanged. So that means there is going to be more oxygen lost to the surrounding than the one being gained. So I said, as light intensity increases, the rate of photosynthesis becomes higher than the rate of respiration until 55 arbitrary units. And after that, some other factor is limiting the volume of oxygen used. That is why the graph kind of flattens. Down here they say the volume of oxygen released by Chlorella is the difference between the oxygen produced by photosynthesis and the oxygen taken in. Use the graph to calculate the volume of oxygen produced in 5 minutes by photosynthesis at light intensity of 50 arbitrary units. To take you back, At 50 arbitrary units, we can see the volume of oxygen produced is going to be that plus this difference here. This part here. So this is 10 plus the 38, that is going to be 48 millimeters cubed. So let's go back to the previous page here. So that's why I got 48 millimeters cubed. You can see it here, and that is why it's my answer. You add up the 38 and the 10 below the zero line. Describe how hydrogen carbonate indicator could be used to investigate the effect of light intensity on carbon dioxide exchange by Chlorella. Now, again, we have to know that we will use this hydrogen carbonate indicator. However, if the concentration of carbon dioxide is lower, it is going to show a yellow color. And if the concentration is high, we will see a purple color. So keep that in mind. However, when we carry out this experiment, it has to be a fair test. We will have to use the same species, it has to be the same mass, all the other conditions have to be maintained, and the temperature has to be maintained. So the only thing will be varying is the light intensity. So I said, set up experiments with the same species and mass of Chlorella in the same concentration of hydrogen carbonate indicator at the same temperature and for the same period of time. Then we are going to use a lamp and a measuring tape to vary the distance of the lamp from the Chlorella experiment. If the indicator turns yellow, then carbon dioxide concentration is low, and if it turns purple, then the concentration of carbon dioxide is high. The key thing here is to ensure that all the other factors are going to be kept constant except the independent variable in order to get results that can be dependable. This brings us to the end of question 3, as well as to the end of this first part of the paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.